Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on Earth, and that's biology. So thank you so much for joining me on this new beginning series. So today we are talking about your biomolecules, the last lecture in your biomolecules. So we'll finish off. So today we are going to talk about enzymes. So before that, all that I've covered, so I'll give you a quick recap. So probably we stopped in metabolism. So what is a metabolic flux? And today we are moving into the concept of enzymes. So it's very short, so I'll finish off enzymes today. All right. So I also hope you guys had an amazing weekend. So you guys celebrated Holy well, because we did not have a lecture on Monday. So it's been a long time since we met. So I hope you guys are doing well. All right. So let's move forward. So we were talking about what is metabolism and what is the steady state or equilibrium state in a living organism, right? So we were talking, before that we were talking about the nature of bond linkage, right? So we were talking about this, how in a polysaccharide, the monosaccharides are linked. So they are linked by the glycosidic bonds. Right? So I hope you guys are able to recollect. Yes, your proteins are linked together by the peptide bonds. Then you have your nucleotides or your nucleic acids. They are linked together by the phosphodiester bonds. So we also spoke about the structure of your DNA. So you have your double helical structure of DNA. Right? So the degree is 36 degrees. So each turn is 36 degrees and the height is 34 Armstrong or 3.4 nanometers. So you have the base pair stacked like this. The distance between the base pair is 0.34 nanometer, and in one particular turn you have 10 base pairs. Alright, so we also spoke about the structure of DNA. So then we went on to talk about metabolism. So your biomolecules have a particular turnover, right? They constantly change from one type or one form to the other. So for example, if I had for example rice, so it is broken down into glucose, and that glucose is used in some other metabolic process. So it's constantly converted from one form to the other form, right? So that is your turnover. And remember, each biomolecule has a characteristic concentration. Like your hormones are always in trace amounts, your glucose is always in higher amounts, right? So they have a particular characteristic value, so that is how they are measured, and that is a particular or typical character that is associated with them. All right, fine. So further, when you talk about metabolism, so your metabolism can be both anabolic as well as catabolic. So catabolic is like climbing down the stairs. So from bigger molecules, they are broken down into smaller molecules, and in this process, energy is released. So anabolic reaction is like climbing up the stairs. So from smaller molecules, larger molecules are being formed. So here energy is used. So for the energy that is being used here, this energy comes from the catabolic reaction. So like you know, the catabolic and the anabolic reactions do not happen simultaneously. Okay, so the energy that comes from the catabolic reaction have to be conserved so that it can be used in the anabolic reaction. So this energy can be stored in the form of high energy bonds, that is your ATP, right? So this is where we stop. So further, so we also know each and every metabolic pathway is highly interlinked, right? So there is a dynamic flow of your metabolites. So the product from here goes into this cycle, this pathway, etc. So it is highly dynamic. So you have the influx and the efflux of your metabolites. Then, irrespective of how many hour reactions that you have or that is happening inside your body, yes ma'am, a very happy good evening. So thank you so much for joining my lecture this evening. So please do like, share and subscribe. So irrespective of how many ever chemical reactions or metabolic reactions that is happening in your body, so every single reaction is catalyzed by an enzyme. So enzymes here are your biocatalyst. Right? So each and every small step in your body which has your metabolic reaction is catalyzed by enzymes. So enzymes are biocatalysts. They enhance the rate of your reaction. So be it the normal step of dissolving your water in your carbon dioxide in water, so that is still catalyzed by enzymes. So today we want to talk about this powerful topic of enzymes. All right, so we are going to talk how the enzymes enhance the rate of a reaction. So what are the factors that is affecting your enzymatic reactions, or what are the classifications of your enzymatic reactions, and what is the structure of an enzyme. So these are the concepts that we'll be talking now. Okay, so yeah, so we'll be talking, or we'll be comparing your enzymes to a chemical reaction, how it can happen. So then we'll move forward to talk about how it brings out high rates of conversion. When I say each and every metabolic reaction is catalyzed by enzymes, so what is so special in enzymes, and how do they bring about this change later, right? So nature of enzyme action, how do they work, right? So what are the factors that affect your enzyme activity? Then you have classification and nomenclature of enzymes. So with this will be done with your biomolecules, so we can have a question session next. So we'll discuss the questions in biomolecules, and then we can have a Menti quiz. So enzymes are proteins except one. So except your ribozyme, everything is your proteins. So almost all your enzymes are proteins, but there are certain nucleic acid which can act as enzyme. Example is your ribozyme. So ribozymes are the only enzymes which are nucleic acids that come from your RNA. Remaining all your enzymes are made up of proteins. An enzyme has an active site, that is the binding site for the substrate. For example, when I write a reaction like A gives B, and this is catalyzed by an enzyme C. Say for example, C is the enzyme here. So the structure of C can be something like this. So this is called as the active site of the enzyme where the substrate A can bind. So when I say A gives me B, so A is my substrate in chemical terms and B is my product, right? So this A binds to the enzyme at a particular site that is called as my active site. So here you have to understand an enzyme can have more than one active site. Okay, clear? Fine. So see, this is my enzyme. So here I have two crevices or pockets, so that is my active site. So my substrate binds into this active site and finally it gives me two products. So that is how it works. Okay, fine. So enzymes are really powerful. So let me tell you how enzymes are really powerful. So if you remember, we are done with your human physiology, right? So in a respiration, so we spoke about how gases are being transported, right? So we spoke something called as a chloride shift, or the hamburger's phenomenon. So what does this say? So carbon dioxide, which is present in your RBC, in the presence of water, it forms your carbonic acid, which is reversibly converted into bicarbonate ions, right? So this conversion happens in the presence of carbonic anhydrase. So when this enzyme carbonic anhydrase is present, you have millions of carbonic acid molecules being formed. Okay? Yes? When this enzyme is absent, you have only 200 molecules formed. So you have a 10,000-fold increase when there is an enzyme. So enzymes are really powerful, and this is one mind-blowing example for the same. Hi W, yes, a very happy good evening. Yes, I'm really good. How are you? Thank you. So please do like, share and subscribe. So once again, thank you so much for watching my lecture, everyone. Okay. Right, so now we're going to see the properties of enzymes here. So enzymes are remarkable. They are highly specialized proteins. For example, example, if I say I have a reaction A gives B in the presence of C, so C is my enzyme, right? So A is my substrate and B is my product. Now I say I have another reaction, P gives me Q in the presence of R. So R is my enzyme and P is my substrate for R, and this is the product. So even by chance, C will not bind to P. Though this is also a substrate, this is also a substrate, C will not bind to P or R will not bind to A. They are very specific for the substrate to which they work. So they are highly specialized proteins. They have extraordinary catalytic power because with the enzymes you saw the amount of molecules that have been produced, right? So they have extraordinary power. So they have high degree of specificity for the substrate, right? So they accelerate the chemical reactions tremendously, of course, to 10,000 folds, right? Yes, probably day after. So today, Monday, you will have the mentee of this chapter, because Friday I want to discuss some questions from this chapter. So we'll have the critical thinking questions discussed in this chapter on Friday, and Monday we'll have the mentee quiz. All right. Yeah, so they accelerate the chemical reactions tremendously. So they are found in aqueous conditions in case of mild pH and temperature, because throughout your body you have diverse pH, right? So like your stomach has acidic pH, your intestines have an alkaline pH, your saliva has an alkaline pH. So each enzyme has an optimum temperature and pH, right? So when you talk about a chemical reaction, so chemical reactions have two aspects to it, right? So either the physical state changes or only the bonds change. So for example, when I say water is getting converted to ice, the physical state changes, right? So from the liquid state, it goes into the solid state, right? So otherwise, if I am talking about a chemical reaction, say for example calcium oxide, you have sulfur, so here you have only the change of bonds, right? So that is a type of reaction that happens. So any reaction that is catalyzed has a particular direction, right? So if A is getting converted into B, either it is reversible or it is irreversible. So every enzyme catalyzed reactions have a particular direction. And the rate at which it formed, in what time it is getting formed, is very important. So when I say W is able to finish 2 chapters in how many days? So whenever with respect to time you talk, that is your rate. So the rate of physical or chemical process refers to the amount of product formed per unit time. Rate doubles or decreases by half for every 10-degree change in either direction. So whether it goes up by 10 degrees or reduces by 10 degrees, the rate actually decreases by half. Okay, right. So the power of enzymes is incredible, because I told you here, water plus carbon dioxide gives you carbonic acid, but in the presence of carbonic anhydrase, you have millions of molecules produced. So in the absence of this enzyme, you have only 200 molecules. So look at the power of enzymes, they are incredible, right? So thousands of enzymes are there which catalyzes unique enzymatic reactions in your body. So they are multi-step chemical reaction. Each step is characterized by the same enzyme complex, right? So your glucose getting converted to pyruvic acid. So this is your glycolysis. This is your 10-step process. And here you have an array of enzymes which catalyzes this process. So how does this enzyme actually work? So you have the enzyme. So first the enzyme binds with the substrate. The substrate binds to the active site of an enzyme. So this is the first step. Once when it binds, so it forms the enzyme substrate complex, right? So first the substrate binds to the active site of an enzyme and it forms the enzyme substrate complex, and this complex is unstable. So that is why I have written within brackets, okay? And this is present only for a transient state. So this is not present for a long time but only a short period of time, right? So very soon it changes into enzyme product complex, and later your enzyme plus product comes out. So this is how your enzymes work to catalyze a particular reaction. All right. Clear? So this is what is explained here. So the substrates bind to the active site. When they bind, there's a conformational change. So they form the enzyme substrate complex, which very quickly changes into enzyme product complex. So this, when it binds, this is enzyme substrate complex, right? So here the bond is getting broken. So when the bond is getting broken, it's enzyme product complex. So finally you have your products and your enzyme coming out. So the enzyme has to come out, right? So now there's something called as a concept of activation energy. So what is this activation energy? So look into this graph properly. So whenever you have a graph in biology, make sure you see what is the x-axis and y-axis. Because in a lot of times you have picture based questions, right? So they will give this as A and this is B and ask you what is x-axis and y-axis. So wherever you see a graph, so graph will always accompany you. If you're going to do engineering or if you're going to do biotech, if you're going to do life science, graph is a must, is a part and parcel of your topic, okay? So whenever you have a graph, please learn to look into the x and the y-axis, okay? So this is our y-axis and this is our x-axis. Right? So x-axis is the progress of the reaction, how the reaction progresses to form the product. And y-axis is the potential energy, the energy that is being utilized. So if you see, there are two curves here. So you have a blue curve and you have a red curve, right? And the blue curve says activation energy with enzyme, and the curve within the red arrow is your activation energy without the enzyme. So from these two curves, you can understand when there is an enzyme, the activation energy is less. Okay, so you represent activation energy as E suffix A. So this is how you write activation energy. So what is this activation energy? So activation energy is always less for enzyme catalyzed reactions. Okay, but what is this? So when the substrate binds to the enzyme, right? So it forms your enzyme substrate complex. The energy of the substrate in the ground state to that in its transition state is your activation energy. So initially you have your substrate that binds to the active site of an enzyme. So this substrate has some amount of energy with it. Yes, when it binds to the enzyme and it forms the enzyme substrate complex, it has another amount of energy. Now the energy here and the energy here differs. That difference is your activation energy. When a particular reaction is catalyzed by an enzyme, the activation energy decreases because they are moving forward to the formation of a product. So when there is no enzyme, the activation energy increases. What is activation energy? The difference of energy of the substrate in the ground state to that in the enzyme substrate complex is your activation energy. So when an enzyme binds, the activation energy decreases. When there is no enzyme, the activation energy increases. Okay, this is the concept of activation energy. So first the substrate binds to the active site of the enzyme, fitting into the active site, right? So the binding of the substrate induces the enzyme to alter its shape. So there's a conformational change in the enzyme, thus forming the enzyme substrate complex, right? After that, it forms the enzyme product complex, then the product and the enzyme comes out. Okay. So there are factors that affect your enzyme activity. So what are the factors? So factors affecting enzyme activity. So there are three factors. First is your temperature. Second is your pH. Third is your substrate concentration. So for PCR, why is that we are not using our own DNA polymerase from the human? Why are we using Taq polymerase from Thermus aquaticus? Because our enzymes are temperature sensitive. Our enzymes can work only at our body temperature of 37 degrees, right? So every enzyme has an optimum temperature. So every enzyme works well at a particular temperature only. So when I say this is temperature, so they work well at a particular temperature only. So similarly, every enzyme works well at a particular pH. When I talk about the enzymes of the stomach, they will work well only in the acidic pH, right? So every enzyme has an optimum pH and an optimum temperature. This you should remember. Alright. So I guess I have the graph. Yeah, pH and temperature. So enzymes function in a narrow range of temperature and pH. Not all enzymes work in all pH and not all enzymes work in all temperature. Okay, so each enzyme shows its highest activity at a particular temperature and pH. So that particular temperature, so this temperature, so this pH is the optimum pH, this temperature is the optimum temperature for that enzyme. Alright, so activity declines below or above the optimum pH and temperature. Right? So low temperature actually preserves the enzymes temporarily in the inactive state. So you store something in the fridge because lower temperature stops the metabolic activity. So I can take my enzyme and preserve it in a deep freezer because low temperature preserves the enzyme in the inactive state. But high temperature denatures my enzymes because my enzymes are basically proteins. So these are very important. Alright? Okay, so the third one is the concentration of substrate. So increase in substrate concentration increases the velocity of a reaction. See, if I have 50 molecules of enzyme and if I have 50 molecules of substrate, so now all the 50 molecules of the substrate and 50 molecules of the enzyme will bind. Right? So now when everything is bound, the velocity of the reaction will be high. The speed at which the catalyzation is happening is high. Yes, the catalysis is happening is very high, right? So when I have more substrate concentration, the velocity of the enzymatic reaction increases. So you would have seen a particular graph like this. I guess the graph is in the previous slide. Yeah. So when there is more substrate, look in this graph. So this is velocity of the reaction versus substrate concentration. So when I have more of substrate concentration, the velocity of my reaction increases, right? So the maximum velocity a reaction can attain is my Vmax. So when I have more substrate concentration, the velocity is high and that velocity is Vmax, right? Now, if I want to know what is the concentration of substrate at which the reaction attains half maximal velocity, that is half Vmax, that is my Km value. See, suppose in a lot of gene therapy techniques, so there is a defect in the enzyme, right? So I cannot just give, so if I want to treat a person with who has a defective in the enzyme, so I cannot give him any amount of enzyme. I should know which concentration of enzyme to give, how much quantity of enzyme to give. For that, this is important, right? So if there is more substrate concentration, the velocity of the reaction increases. At the same time, I should also know what is the concentration of the substrate that can give me half maximal velocity. So probably this is my half maximal velocity. So I draw a line and find out the concentration. This concentration is my Km value. So Km is my Michaelis-Menten constant because he was the person who discovered this graph for us. Alright. So this is the concentration of substrate and this is how it affects us. Alright. So next comes a concept called as inhibitor. So how does inhibitor affects a particular enzymatic reaction? So I will look for a blank slide to explain this, right? So suppose if I have a substrate like this, okay, so this is my substrate and this is something identical to my substrate, but this is not my original substrate. My substrate is S and this is my copy, that is my substrate S dash, okay. So now my enzyme is like this. This is my enzyme and this is the active site of my enzyme. Now this can also fit, this can also fit, but the enzymes are specific to their substrate. Only if this binds here, the enzyme will function. If this binds, the enzyme will not function. So this is called as an inhibitor. The one which inhibits the particular action. So this inhibits the action of an enzyme. So this is an enzyme inhibitor. Alright. So this is a type of competitive inhibitor. This is competitive inhibitor because this is similar to the structure of the substrate. This is competing with the substrate to bind to the active site, right? So this is the first type of inhibition. So example can be your succinate and malonate. So succinate is the original substrate and this is our inhibitor. So they both compete for the active site of your succinate dehydrogenase enzyme. So succinate dehydrogenase is an enzyme. The active site actually binds to succinate, but malonate also has the same structure. So when malonate binds, the enzyme does not function. There's another type of inhibitor that is called as allosteric inhibition or feedback inhibition. Alright, so the first step in your glycolysis, glucose to glucose 6-phosphate. So this is catalyzed by the enzyme hexokinase. So when you're feeling very hot, you turn on the fan, you turn on the AC. After some time you feel cold, you turn off the AC, right? So similarly, when the product is formed in large amount, when glucose 6-phosphate is formed in large amount, so this glucose 6-phosphate inhibits this enzyme. So this is feedback. So whatever output you're getting, that itself will inhibit the process. So when glucose 6-phosphate is more than that is needed, so this glucose 6-phosphate itself will come and inhibit the enzyme hexokinase. Alright. Clear? So these are the two types of inhibitors. So we'll stop here. So we have two more parts to this concept of enzymes. One is nomenclature of enzymes. The other one is what are cofactors. So anyways, Friday I am discussing critical questions from this chapter. So I will explain these two parts on Friday. Alright, so I hope you understood the basic concept of enzymes and how enzyme works to catalyze a particular biological reaction. Right? So I hope you enjoyed my class and please be here on Friday at 4:30 p.m. So until then, this is me wishing you a very a beautiful days ahead. So please take care and stay tuned. Thank you.